really think Thai anthropology is an exciting, cutting-edge place to be thinking about the human. And the theme of the conference on hope, on anxiety, on uncertainty really speaks to some of the core concerns in my own work, but I think related to the field in general. So in some ways today, I'm going to tell you a relatively simple story. So if you remember anything from my talk today, it's because there's viral particles in your brain that are getting excited about what I'm saying. So I'm gonna walk you through that story a little bit later. But this talk is also thinking with viruses as a strange figure of hope, an unexpected figure. We've all just lived through a pandemic where the coronavirus, SARS-CoV-2, profoundly disrupted the modern world system. So in part, what I want to do is a little viral therapy here, trying to teach you how to live with viruses, and also maybe how to be happy with viruses in our midst. And I'm using happy here as a theoretical word in conversation with Sarah Ahmed. So in English, if you look at the key word, the origin of the word happiness, it means being full of hap. Hap is about contingency. So in English, we also have the word happenstance, or hapless means someone who doesn't have a lot of opportunities. So being full of hap or being happy means living in contingency. So I'm going to try to unpack human nature. This is going to be a talk that's fundamentally about an anthropological question. What does it mean to be human? What does it mean to be alive? This is a talk that will also engage with themes in the environmental humanities. How do we live with the uncertainties of the Anthropocene and form contingent coalitions with viruses to navigate the uncertainties of these times? It's also a science and technology studies talk, an STS talk. So thinking about human viral futures with new kinds of gene therapies. So kind of those three themes are going to be embedded in the talk today. Let's see if this works. Yes. So in 2008, Donna Haraway published a very influential book. It's called When Species Meet. And at that point, the role of viruses in the human condition or in human cognition wasn't really well known. Hadn't really been studied yet. So she talked about the human as a multi-species assemblage. She insisted we have never been human, and she used musical metaphors to understand what it means to be human. She talked about the human as a coordinated symphony, sort of an orchestral production. And one of the facts that she was excited about related to bacteria, that there were more bacteria cells in the human body than human cells. So that's in part what motivated this intervention. Anna Singh, and both Donna Haraway and Anna are my mentors, so I'd like to honor them today. Anna Singh, in a related gesture, talked about human nature as an interspecies relationship. So I want you to hold on to those two broad ideas about the human condition as you listen to this talk. Building on their work, my new book, which is forthcoming with Princeton University Press, suggests that human nature is viral. So this cartoon here is meant to illustrate the human genome. So, our human DNA is packaged on chromosomes. We have 24 of them. But a little known fact emerged in the year 2000 when they did the Human Genome Project and sequenced the entire human genome. They found that about half of the human genome, half of our DNA, is viral in origin. So there's many different kinds of viruses and viral remnants, ancient viral infections that might have lost some of the key viral genes that make us who we are. The bottom half of this is sort of the human part of our genome. About 55% of us is human or primate, but the rest of us comes from these viral infections. And I'm going to get into one of those viral infections in some detail. So the one that I'll talk to you about that makes us think is called ARC. I'm going to slow down later and really explain what this virus is all about and how it makes us think. but broadly, I want you to think, in that video that's playing in the background, picture that as the inside of your brain, and all those little particles that are floating around are active viral infections. So ARC or ARC is one of those viral infections that we have very good stories about. But it turns out that there's a multitude of viruses in our brain. So parts of the body, so when we had the microbiome findings from about 15 years ago, they were focused on the microbes in our gut. They thought that the rest of the body was sterile, that there weren't, so there's no bacteria in our blood or in our brain, but viruses are throughout our body. They're throughout our blood, throughout our cerebral spinal fluid, throughout our brain. So I'm going to think with that fact to try to undo conventional understandings of human nature and give you new tools for thinking about yourselves and thinking about what it means to be happy, full of hap with viruses. So I'm also not only thinking with viruses today, but thinking with friends. During the pandemic, this was my main activity. I had a Zoom room every week where we were reading papers together about coronaviruses. So these are some of the many scholars that came to this group. So every week we would read a new paper and discuss it. Among the key authors that I'm going to engage with today are Hannah Landecker, Johnny Turnbull, Tony Hatch, and there's many others here who are very much informing my ideas of this talk and my new book. So panning out broadly, what I want to do is kind of inventory some of the cultural narratives that influence how we think about viruses and think with viruses. One of the influential anthropologists who's already written about viruses is Beth Covenelli. And Beth really represents, I think, the mainstream cultural narratives about viruses. Viruses are horrific. We have, in cultural spheres, we have all kinds of movies about zombies. The zombie apocalypse was one of the narratives that kind of structured how we understood the pandemic. Vampires, you have vampires proliferating in cultural spheres. If we're thinking about politics, I know that we're in a faculty of politics here. Povinelli describes the viral mode of politics as terrorism. But I'm going to insist that that's a very small minority of viruses. So if you think about that cartoon of the human, virologists thought that they had their work done in the 90s, that they knew all the viruses and that they were pathogens. It turns out most viruses aren't terrorists. Mostly they have a symbiotic politics or engage in a politics of symbiogenesis, to continue with one of Donna Haraway's key words. So I want to acknowledge Beth Povinelli as a key pioneer in viral theory, but insist that we think beyond terrorism. So one of the other points of reference in terms of culture here is Bjork. She had an album that came out in the year 2010 on biophilia. And in that album, she's got a song where she imagines herself as a symbiotic virus and imagines her partner, her lover, as the host. My colleague Stefan Helmreich has already done a really masterful analysis of this song and this album to think about the human condition. He wrote a piece about Homo microbius, the human that's composed of microbes. And I'm very much building on his work too today. What I've basically done is taken the queer theorist Eve Sedgwick's work and used this to understand the field of virology. So Sedgwick famously encourages people to get beyond paranoia. So living in these uncertain times, these times of anxiety, it's easy to be paranoid about what the companies are doing, what capitalism is doing, what the nation state is doing, what your neighbor might be doing. Paranoia is a pervasive logic of the early 21st century. But Sedgwick insists, this sometimes is a good way for understanding the possibilities of the world. It is an imagining that is trying to anticipate all the bad outcomes. But she's also insisting that this isn't the only way to know the world. And she's a literary scholar. She was reading queer theory, reading queer literature. And in addition to having these paranoid approaches to thinking about the archive of literature, she suggests that we might engage in what she calls reparative reading. So a reparative reading, or orienting yourself towards the literature in a mode that thinks about repair, is about being generous. It's about being generous, having generous interpretations of key characters in a story. It's about thinking what needs to be done in a community where love and care are kind of central. So it's about thinking relations and how to fix damage, how to fix problems and infrastructures, social milieus, and environments. So basically what I've done in my new book project, and what I'm going to try to do in this talk, is approach the virological literature not with paranoia, not just trying to think about all the bad outcomes that might happen as a result of a viral infection, but to think about reading the virological literature with these reparative strategies from Eve Sedgwick. So in part, what I've already offered is a reparative reading of the human genome. You know, we're mostly, not mostly, but at least a significant portion of us is viral. I'm also trying to reread the human virome. So the microbiome, which got a lot of attention 15 years ago, the microbiome largely is focused on the bacteria that is existing with us and making us human. The virome is the word for the sum total of the viruses, whether those are baked into our genome that we're born with, or more fleeting, transient infections, viruses that come and go. But beyond the human virome is this larger universe of the unknown, the virosphere, all these viral particles that abound around the Earth that we know very little about. So in addition to thinking about the viruses in your brain right now, with this image in the background of all those particles, I want you to think about all the viral particles floating in this room, all the viruses that are floating outside amongst the trees, amongst the animals. And some of these viruses, we inhale billions of particles every day. Some of those are capable of replicating in our bodies. So viruses connect us to other forms of life. I'm also in this talk going to briefly describe reparative strategies for living with pathogens. So I know that some of you in this audience have also written about HIV. It's a very pervasive virus in human populations. It's really one of those viruses that has transformed human social practices, human sexuality. So part of what I'm trying to think about when I'm thinking about the possibility of symbiosis and symbiogenesis, living with these other viruses, is how to transform a pathogen into something else. So I've published a short article. This is a free PDF that you can get online called Impure Hopes. And it describes HIV positive patients who signed up for the world's first gene therapies, they became the world's first edited people. This is before CRISPR-Cas9 became a disruptive and an influential tool in biotechnology. Their genes were transformed, edited with another tool called zinc fingers. Basically, what the scientists were trying to do was repair their cells and make their cells impermeable, resistant to HIV. That experiment failed in some interesting productive ways, but their story is one of how to live with science, how to live maybe in antagonism to certain forms of capitalized science, and also how to live with the HIV virus in a way that might even be considered happy. So part of the story about HIV is a story of technological innovation that lets people live with a virus that was once a death sentence, that if you are able to access either conventional, highly active antiretroviral therapies or these new gene therapies, you're going to have a life expectancy similar to normal. My book also describes how the HIV virus itself has been turned into a tool of genetic engineering for people. So you might approach these kinds of tools with paranoia. Not many people probably want to get HIV. It's not a virus. There are some queer communities that deliberately spread this virus, but for most of us, the figure of HIV is a figure of terror. But I'm trying to show in this book how it's actually been turned into a therapeutic agent. So genetically modified HIV viruses are being used to deliver cutting-edge gene medicine, so transforming the human genome. So this is one way, concretely, that viruses are a figure of hope. Gene therapies are being used to treat blindness. They're being used to treat sickle cell anemia, various kinds of cancer. So this virus that once was the source of anxiety, debilitating medical conditions, taking out your immune system, has become an agent of repair, an agent of symbiosis. So in some ways, this is a story of cruel optimism. Just pausing on gene therapy for a second before I get into how viruses make us think. There's new gene therapies on offer now. They cost about $30 to manufacture. They're not that different than an mRNA vaccine. And think about, the profound public health benefits of these vaccines. But these companies like Novartis that are selling these gene therapies have claimed intellectual property, and they're selling them for millions of dollars. So this particular gene therapy for a rare condition called spinal muscular atrophy, the drug is called Zolgensma. They're selling it for $3 million a dose. And it's a story of, again, innovation and inequality, but it's also a story of trying to sell hope and trying to sell happiness. But in many instances, these profoundly human stories aren't turning out to be the salvation or the miracle cure that are being promised by the companies. So here's one of the children that received this drug. And, many of these children, even after getting these million-dollar treatments or multi-million-dollar treatments, they're living with lifelong disability. They're living with lifelong problems. They're going to the emergency room time after time. Some of them are even dying. So, with this story of innovation and inequality, what we see are new regimes of capitalism that are exploiting hope. So even as biotechnology promises new solutions, new cures to age-old diseases again, blindness, sickle cell anemia, they're using HIV viruses to combat HIV, all very interesting and exciting, but there's this fundamental pathology of power that is taking place with these emerging medicines. So you might think about them with Lauren Berlant, about the cruelness of the optimism that is pinned on this hope. When you get the cure, it's not necessarily what was dreamed for. These children, many of the parents who enroll their children in these treatment regimes imagine this profound transformation, but it very often doesn't manifest that way. Okay, so now I'm going to get to that science and technology studies story that I opened with, and I've offered a written version of this. And so this next part of the talk, if you're curious, I understand there's a Thai version of it available to read. So I mentioned the whole human genome and how about half of us is viral in origin. I'm going to be building on Haraway's claim that we've never been human. I'm going to suggest we've always been viral. So this is, again, trying to repair our relationships and our feelings of anxiety in relation to viral infections and the virus sphere. I'm building on Anna Singh, I'm going to make a case that human nature is more than an interspecies relationship. Interspecies just implies two species, but I'm going to think about how the human and consciousness itself is a hybrid multi-species co-production. And I'm going to walk through what I mean by that fairly slowly. So first, I want you to get beyond the idea that a virus is a particle. I want you to think with this animated video here about viruses as lively or living processes. So if you try to understand the virus as a particle, that's much like trying to understand a tree and all the complicated properties a tree has just by looking at a seed. So what happens when the virus goes into the cell is very dynamic. You could think with Deleuze here about deterritorializing. The virus as a particle is this round sphere, but what you see in this movie, something going all over the place. It's interfacing, it's tracing these different organelles within the cell. It's using the cellular organelles to make copies of itself. Many people think about viruses as just docking to that external receptor in a cell. They don't think much about what happens after the virus gets inside. But I want you to think again about the liveliness of viruses when they become lively in the environment of their host cell. So here I'm thinking with the philosopher John Dupre and the biologist and philosopher Stefan Göttinger. So they again insist we think beyond the particle and think beyond this binary of life and non-life to think about a virus as something that becomes a lively process in the right environment. I also want you to think seriously about the possibility that a virus itself is a thinking being. So if it's lively, it's a question. Can this thing, or is it a thing? Can this process respond to information in the environment? This is what, according to Gregory Bateson, being able to perceive a difference in the world and respond to it is a key criteria of a mental system. He came up with these formal criteria for mental systems. Bateson is a famous anthropologist. He was working in cybernetics. He also studied animal behavior, working across many fields. And he came up with these formal criteria of mental systems. So what I've tried to do in the book is apply those criteria to a very fun text written by Mary Ewell. So if any of you are inspired to think more about viruses after this talk, I really recommend that you go out and buy Mary Ewell's book. It has really great cartoons in it. It really breaks down what a virus is. So she's particularly interested in phage or bacteriophage. That's the kind of viruses that infect bacteria. So the book is titled Thinking Like a Phage. And what I'm trying to do is take seriously that very possibility that viruses think. So again, phage are bacteriophage. That's the kind of virus that infects the bacteria in your gut. You have phage running through your body right now. So again, minds, according to Gregory Bateson, are able to perceive and act upon differences in the world. Mental systems make sense of information. Again, that's differences that make a difference. And in this book, Mary Ewell, a virologist, provided evidence that viruses can indeed sense information, and they use information about the environment to transform their hosts. Here's a very complicated, messy diagram of how that takes place, but you can think about that in relation to that video that I showed you a few moments ago. So I insisted that we're mostly viral, and the one particular kind of virus that I'm going to tell you about today is a retrovirus. So that means it's related to HIV. It's got some of the same basic proteins as HIV, but again, we're born with this virus. It's part of our human genome. One other name, let's see if I can do, yes, okay. So the kind of virus that I'm talking about here is an LTR retrotransposon. Basically, that's just another name for an endogenous retrovirus. That means a retrovirus that is in the human genome. So this is Jason Shepard, and he's the one who figured out this very complicated story about how viruses are involved in making memories with us. So he's pictured here with an artistic rendering of the ARC protein when it's in its viral form. And these are electron microscope pictures of this virus. I'm going to give you a brief history of this molecule to help you understand these claims that viruses make us think. So this protein, before its viral properties were understood, neuroscientists knew that it was involved in memory. So this is one of the first proteins that is activated when a new memory is formed. So this has been known for more than 30 years. Jason did his PhD in Paul Worley's lab at Johns Hopkins, and Worley is one of the first ones to figure out the basics of this story. Early on in the 1990s, they used transgenic techniques to create forgetful mice. So they basically deleted this gene, they knocked it out using genetic engineering techniques, and found that these mice were constantly surprised. So if you use traditional learning behavioral tests with mice, if you knock out the ARC gene, if you delete it, the mice can't remember anything for longer than 10 minutes. So they're basically constantly living in the present. So when Jason was a PhD student in the late 1990s, he used a new bioinformatics search tool. So bioinformatics is basically a fancy name for like Googling a gene sequence. So there was a new search engine that came online in the 90s called BLAST. And what you could do with this search engine was take a gene sequence, enter it into this search engine, push enter, and see other genes that are related. So when Jason did this as a graduate student, he got predictable results. He found that ARC is closely related to similar genes in monkeys and cats and rodents, but he also got an unpredictable result. He found that it was related to HIV and other viruses. But he was a first-year PhD student. He thought, it must be a mistake that, many first-year PhD students don't have the confidence to run with a new discovery. So he forgot about this, and he did his PhD. He focused on other dimensions of ARC, really technical problems, published some papers in leading journals like Cell and Neuron. And then many years later, I believe in about 2012, 2013, he founded his own lab at the University of Utah. And in the early months of setting up this lab, he was training a new laboratory technician named Nate. And Nate didn't really know how to do any of the protocols, so Jason basically had to teach him the very basics. So one of the first things they tried to do together in the lab was just isolate this protein, ARC. So to do that, you put it in a column like this, and he knew how much ARC weighed, so he was looking for it in a lightweight part of the column, or I think that's up here, but he actually found it down there. So it was many times heavier than what he was expecting. So this laboratory technician was also trying to learn how, he wasn't really interested, he was not thinking about viruses, he wasn't thinking about that blast search, he was just trying to teach his technician how to do basic laboratory work. So the technician also wanted to learn how to use an electron microscope. So they took pictures of ARC, and he thought it might be clumping, but he didn't know why or what it would look like. So he got these pictures, which he didn't know anything about what they looked like. To him, it just looked like a messy black and white picture, probably similar to you. He didn't know anything about viruses, but he did give a talk, a work in progress talk, and one of his colleagues was there who was an expert on HIV. And that expert immediately recognized the picture. So this is an electron micrograph of HIV. To us, that might not look that similar, but the expert was able to immediately identify it. And that's when Jason thought back to that blast search that he did as a graduate student. And it was kind of this aha moment where he brought all these different pieces of the puzzle together. And that's when he realized that ARC was a virus, a retrovirus, a relative of HIV that's living within us, that's living in our genome. So the story of how ARC makes memories with us is actually kind of complicated, and it involves basically telling you the basics of how neuroscience works. So a famous mantra from Hebb is repeated here. Neurons that fire together, get excited and send these electrical impulses. They wire together, they form new connections. So ARC is involved in this wiring process. Basically, the excited neuron sends a signal with this viral protein. So here you see, ARC. And the really interesting thing is, it's actually got a gene inside. So that's usually ARC genes. But he also found that ARC, these viral particles, can transmit other genes. And this is really the tip of the iceberg. So if you think back to that picture of the human genome, ARC isn't the only viral infection in our brains or in other parts of our bodies. There's constant signaling processes happening. This is why I'm suggesting we think about human nature as an unstable viral phenomenon. But with this story of making memories, the excited neuron sends these viral particles, and they're received by the neighbor, and they basically alter the synaptic gap. So every time a neuron fires, there's a little gap between one cell and the other, and this is where the memories get formed. It's even more complicated. Take my word for it. But if you really want to go deep into this story, Jason Shepard has a really lovely TED Talk that kind of explains the nuanced details from a neuroscience perspective. But for me, thinking back about some of these fundamental anthropological questions, not only is this key to the ongoing memory formation, but this is kind of critical to the human condition. It's something we share with primates. We share this viral infection with birds, not fish. Fish make memories in different ways. I think they're dimmer than our memories. But interestingly, this virus, or a relative of this virus, has independently infected insects. So insects make memories in similar sorts of ways. I think for me, it's a figure of multi-species connections. But it's also a way of thinking back about, how do we rethink historical consciousness? Some theorists of consciousness have talked about literary histories and how someone's living hand to mouth if you're not aware or conscious of thousands of years of written history. I'm suggesting that, if we're not thinking about millions of years of viral symbiogenesis, these long-term alliances, these long-term symbiotic connections we've formed with other kinds of life, then really our whole project of historical consciousness, the whole modern project, wouldn't exist. So this is a really core aspect of who we are and enables all of the possibilities of, the human condition that are so elaborate, nuanced, and complex. So now that I've told you kind of a hopeful story. So I've told you initially an ambivalent story of hope, innovation, and inequality in gene therapy. I'm trying to get you to rethink the human condition, thinking about being happy with the viruses that produce this unstable, dynamic sort of human organism. There's many other viral stories I could tell you. Like there's other viral infections that shape the architecture of our brains. This was just about kind of the contingent connections that we form with viruses and memory. Now I want you to get anxious again and remember what it was like to live through the coronavirus pandemic. So this is a scanning electron micrograph of a coronavirus. But I also, in bringing back those unpleasant memories from the pandemic, I'm going to invite you to rethink them with what you've just learned about viruses and imagine the possibility of being happy with viruses in our mix. So again, not just viral horror stories today, a la Beth Povinelli, but stories of possible coexistence and peace. So Alexander Sasha Gorbalenya is arguably the world's foremost authority on coronaviruses. He described the first coronavirus genome in 1988. This is before the human genome was characterized. And one of the things that I like about Sasha is not only his deep knowledge of coronaviruses. I think of him as kind of a natural historian of coronaviruses, someone who's genuinely curious about what these viruses do in the worlds where they live. But he's also a philosopher and theoretician of how the human condition is being transformed by viruses. So he gave an oration. So he was born in the Soviet Union, fled communism as soon as the Berlin Wall fell. He worked in the United States for a long time. Then he took up a post at Leiden University, where he is currently professor emeritus. And in a oration at Leiden, a distinguished oration before he retired, he gave a lecture with this title, "Can We Live in Peace with Viruses?" And here he's reflecting on the fact that we're all infected with viruses. All of us right now, in addition to those latent infections in the human genome, we're harboring other kinds of infections that are more transient. Of the billions of particles we inhale every day, some of them are capable of replicating in our cells. Some of them are producing changes in our bodies. And he wants us to think about these infections ecologically, to think about how they link us to other forms of life, and to get beyond this image of the virus as a terrorist, to think about possibilities of coexistence. So in relation to these infections, he says, virology in many ways is in its very early days. We don't know what most of these viruses are doing. So viruses that don't produce major disease, we don't know. Are they bad? Are they good? Are they something in between? And he's insisting that before we use this logic of eradication, we think about, again, viruses in an ecological context. I also want to introduce you to Linfa Wang, who is very good about thinking about viruses, in particular coronaviruses, in ecological contexts. Linfa is currently the head of a major research unit in Singapore. He's also got an interest in a company called Paratus, both his Prepare Institute in Singapore, that has about $100 million budget, and this company he's involved with called Paratus also has about $100 million budget. But what he's really known for is the hypothesis that bats are reservoir hosts of coronaviruses, specifically of SARS coronaviruses. So this is a picture of his 2005 paper in Science that first puts that hypothesis forward after the original SARS outbreak. So with Sasha Gorbalenya's knowledge about coronaviruses broadly, and Linfa Wang's work about bats and coronaviruses, I first came to Thailand in 2022 investigating these questions here. I was interested in places where coronaviruses are actively circulating amongst animals and people. And from a theoretical point of view, I really wanted to understand how a virus that is symbiotic in bats, a virus that doesn't make bats sick, became a pandemic disease in people. Is this a question that's best answered in relation to properties of the virus, or something more complicated? So for starters, this is the kind of bat that carries coronavirus, a horseshoe bat. The genus is Rhinolophus. And here's a picture of SARS-CoV-2. So thinking back to Sasha Gorbalenya, one of his big points is that it's a mistake to confuse the virus with the disease. So there's HIV, which is a virus. There's AIDS, which is a disease. So most people with HIV these days who are on medication don't get AIDS. So similarly, if we think about SARS-CoV-2, many people contracted this virus even before they got vaccines and didn't get seriously sick. So part of the question is to think about, how to distinguish, again, the virus, SARS-CoV-2, from the severe respiratory disease, COVID-19. So I'm going to ignore Wuhan today, but if you want to read my book, which I don't have available yet, it's going to, academic books take forever to publish, so it's going to at least a year or two away from getting the book. In the book, I visited Wuhan and interview all the key people, including, the head of China CDC, people associated with the Wuhan Institute of Virology. I basically conclude that this virus did not originate in Wuhan. And even, if we think with Foucault, if we think with Nietzsche, the very idea of an origin story, this outbreak narrative that is invested with so much political valence. But, ultimately I found the narratives about where this virus came from to be profoundly orientalizing, using the language of Edward Said. Said talks about how, in the so-called Orient, there's so-called exotic animals, exotic food markets. I want you to forget all the outbreak narratives that you were taught during the pandemic, to think about biogeography in a really complicated way, to not just think about Thailand. So there's 49 kinds of horseshoe bats. And why I'm focusing on horseshoe bats is because Linfa Wang and others have demonstrated that horseshoe bats have the most coronavirus biodiversity in the wild. So one of these species, the greater horseshoe bat, ranges from Japan all the way through the Middle East, through Europe, all the way into southern England. We don't know where these viruses were spilling over. We've got some pretty good guesses. And this is a picture of Southeast Asia that shows just patterns of horseshoe bat biodiversity, where most species of these bats are found. Here's a closer zoom in, and this is dealing with human blood samples. And these are blood samples that were taken before the official outbreak was reported in Wuhan. So what we have is in Thailand, up in Chiang Rai, up to 18% of the population had antibodies to coronaviruses before December 2019. So there's a pattern of pre-pandemic exposure. And if you aggregate all of those blood samples that were taken before the pandemic, and you do some math, like Lin Fa and his colleagues did, you find that every year about 60,000 people were infected with animal coronaviruses in Southeast Asia. And again, we don't necessarily need to think that Southeast Asia is the only place where these spillover events were happening, but we've got a very concentrated history of research here in Southeast Asia, which is in part what led me to come to Thailand. So in part, what Lin Fa and others who have analyzed these historical data point to is the fact that there's actually some benefits that you can get from being exposed to an animal coronavirus. To be clear, I'm not recommending that you go cuddle bats today or pick up, street cats or other kinds of animals that might have wild viral diversity. But many people, especially in this paradigm of modernity that aims to separate nature from culture, that aims to eliminate certain kinds of multi-species relations, especially in rural hinterlands, these kinds of findings go against that modern project, which is a project of biosecurity, policing boundaries. So in fact, some kinds of multi-species encounters that have viral dimensions can actually produce good probiotic benefits for people. So to be clear, probiotics is my own analysis of the situation, but Lin Fa and colleagues describe very clearly how these kinds of viral exposures can give you antibodies. So if you were exposed to a bat coronavirus before the pandemic, that might have given you some resistance to SARS-CoV-2 and prevented you from getting seriously sick with COVID-19. So I've published a preliminary analysis of this with some colleagues here in Thailand. So if you want to think about being happy with viruses, or in the idiom of this paper, how to think about luck in relation to viral exposures, this is also a free article that you can read on the internet. But my latest work is departing from that earlier analysis of luck and happiness to think with Karl Marx and other theorists about the broader context in the Anthropocene, in this time of great anxiety, in this time of great uncertainty, how the food system and what Marx calls metabolic rifts are part of what's making us sick. I'm interested in how this might be damaging individual animals, bats, and also be creating opportunities for viruses to jump across species lines. So right now we're in a metabolic moment. These theories about metabolism are quite influential these days in the humanities and the social sciences, fields like geography. Here are some of the key thinkers. So you've got kind of the thinkers up here that are trying to create an ecological Marx, and we could probably argue whether or not Marx was an ecological thinker. I think many people have argued that nature is treated as something that's quite instrumental in the original Marxist texts. But we've got a new generation of scholars, including Anthony Ryan Hatch, Hannah Landecker, my close friend Johnny Turnbull, who are rethinking metabolism in molecular ways, in addition to these classic Marxist ideas. So classically, Marx describes how the food system has produced what he describes as a metabolic rift, a break in these energetic processes. If you read Marx through the Japanese thinker, Kohei Saito, Particularly, there's a focus on excrement, on poop, in the original Marxist text. Marx was very worried about this event that happened in 1858. So he describes prior to that moment, there was kind of a cycle. I think in the 20th century, we would use the language of homeostatic circuits to think about how things stay stable. This wasn't how Marx was thinking about it. There was a very peculiar language for thinking about disease in those days. This nebulous idea of miasma, basically smelly air. They thought that that produced diseases like cholera. Not true. So it turns out that cholera is mostly, a waterborne disease. But part of what he was trying to think about is how these environmental problems produce human disease. And to a certain extent, he did describe something that is accurate. Part of what they were doing in the 19th century was importing guano, importing excrement to try to boost agricultural production. So this resulted in a lot of the importation of bird and bat guano from colonial territories. And part of what you could think about in relationship to metabolism is how life itself was working. So if Marx was trying to describe these metabolic processes that were taking place on a planetary scale, in the book, I go into quite some depth into how to think about metabolism in relation to bats, and also how the modern agricultural system is damaging bats, damaging bat health, leading to moments where they're literally falling from the sky, flapping on the ground. Part of this might relate to just the ways that deforestation has made bats hungry. Many bats live for a long time, so many of them probably have the historical memories of the landscapes where they forage being transformed from forest into plantation. But there's also probably some acute pesticide exposures that are damaging their bodies, leading to viral shedding. So again, I'm thinking with friends here, and in part, I'm wanting you to think not only about what's happening with the plantation economy in places like Thailand and Southeast Asia, places where bats are getting injured, literally falling from the sky, shedding viruses, but I want you to be thinking about consumers of sugar on the other end of the modern world system. So Tony Hatch has written about metabolic diseases that have proliferated with sugar. He's writing as someone who has a metabolic disease himself. And early in the pandemic, he was writing in a pessimistic register about how unlikely it was that him and people like him would survive. One of the other papers that we read as part of this reading group was a metabolic handbook for COVID-19. And to very quickly connect the dots, basically, if you have metabolic disease, if you have type 2 diabetes, your body is making more ACE2 receptors as a way of protecting yourself. So if you have a metabolic disease, this is helpful. This helps your body live with elevated sugar. But if you get infected with SARS coronaviruses, this is going to give the virus a better way of proliferating with your body. So I'm going to wrap up quickly here. In short, I want to suggest that bat guano might actually help repair metabolic rifts. There's been some great research actually done here at Chulalongkorn University by Ajang Suprapon. And I believe that there's one member of her lab here today, Bo. Together, they've published an amazing paper in Viruses, which is one of the flagship journals of the discipline, basically studying whether or not it's risky at these sites of guano collection for viruses to jump into people. In short, they found, after collecting blood samples from a whole lot of people involved in guano collection, that it's very rare for these viruses to jump directly from bats to people via guano. So this is a relatively safe, biomedically safe activity. And I think we might reflect and step back from a lot of concern from biomedical scientists in the One Health paradigm to think about how agricultural production that's using synthetic pesticides and fertilizers might be repaired with guano. So thinking with Karl Marx, maybe it's time to make, and Donald Trump, maybe it's time to make guano great again. So I'm going to just briefly conclude with two questions. So first, I'm thinking with Donna Haraway about symbiogenesis. She's thinking of symbiogenesis as this looped, braided, outreaching, involuted process. I want you to think again with these viruses that are throughout our bodies. Haraway used symbiogenesis not only in a biological register, but in a social register to think about how AIDS activists entered into symbiosis with capitalism. So if I had more time, I would tell you about how the guano collectors here in Thailand are making these symbiotic articulations with capitalism to distribute guano in organic agriculture. I see a real parallel between the strategies that AIDS activists used in the 1990s and some of the agricultural production that's happening here in Thailand today. I want you to think beyond the intellectual property regimes that are resulting in innovation and inequality with gene therapy, and think with some of the innovative work that's happening in biohacking. Lilia Rani has an amazing book about hacker cultures and innovation in India. And here in Bangkok, there's the Fab Lab that's located nearby that is starting to use some of these tools of biotechnology to innovate within this space, to imagine modes of innovation that aren't about repeating these very drastic regimes of inequality. So for me, symbiogenesis not only involves these biological thinkers that Haraway cites. In my book, I'm going much deeper to think symbiosis. I actually found in some Greek texts that this might actually be considered some originary queer theory. I found some symbiosis in some ancient Greek texts. I also found symbiotic practices happening where good viruses were being spread by traditional medicinal practitioners in China centuries before the vaccine was discovered in Europe. I also want to think about these future possibilities of viral symbiogenesis. And this is in conversation with some of my work on gene editing in the Mutant Project, to again think about those biohackers that might be doing biotechnology otherwise, but also to think about how we might undergo a radical transformation of the food system, to think about living in happiness and harmony with viruses on a landscape scale as well. So this is my concluding question. Can viruses help us think about the possibilities of symbiosis with capitalism? So thank you very much.
โอเคครับในลำดับต่อไปนะครับขอเชิญอาจารย์จิราพรขึ้นมาดำเนินรายการต่อไปแล้วก็เข้าสู่ช่วง Q&A นะครับซึ่งนะครับแล้วก็เมื่อเข้าสู่ช่วง Q&A นะครับผมขอเชิญท่านผู้เข้าร่วมการประชุมทุกคนนะครับสามารถถามคำถามถึง Keynote Speaker ของเราอาจารย์ Eben ได้ในวันนี้นะครับซึ่งท่านสามารถถามได้ทั้งเป็นภาษาอังกฤษและภาษาไทยซึ่งจะมีอาจารย์จิราพรเป็นคนแปลนะครับผมขอบคุณครับ
Thank you, Urban, for such a very thought-provoking. I think along with the virus, and I live with the virus. Actually, also as a social scientist, like I will bring the knowledge that I was study in the early high school about biology, right? Reading with you, your work, and also listening to you, right? And also, I think among the Thai people that we haven't think about it. We live with the gono, or we spreading the gono in our garden, in our field, and many things. So that actually kind of very much relate to us more than we think about virus as a disease, but something that we live with it in everyday life. We breathe in, we breathe out, or in every dimension of our body. So I think from here now, I guess there's some people, some veterinarian that I saw, some virologists maybe, and other doctors, or actually the social scientists, we can ask the question. มีคำถามไหมคะ
Thank you for your presentation. I'm Thanapat from University of the Ryukyu, Okinawa, Japan. Actually, about virus is all Greek for me, but your presentation provide really interesting information. Actually, as a sociologist, one subject I'm teaching is about multicultural symbiosis in Japan. We need to teach Japanese students how to live with a foreigner. So what I'm thinking about virus, I interested in how can human interact or be interacted with virus without using microscope or without infected. I'm thinking, for example, I got flu and maybe I can talk to virus that, let's cease fire. I will stop taking medicine, so you stop attacking me. So my question is, for sociologists, in a society, sometimes people around us is kind of like a virus because can make us sick. Just human and human interaction is not easy. It's too difficult. And how come we can interact with something it cannot be seen, but we can feel in body? So my question is, I want to hear your opinion, how to interact and to be interacted with virus. Thank you.
Thank you for that question. And so I think it's super interesting that you're teaching symbiosis in this cultural realm. So I'd like to think about symbiosis or symbiogenesis, the process of creating new symbiotic relations, as a transformation of relations of antagonism and discord into harmony. And there's lots of ways of thinking about how to do this. The one that I keep going back to is HIV, and if you look at those early antagonistic dynamics that AIDS activists had with Wall Street, with capitalism, with the U.S. government, and basically politics of kind of headbutting and fighting, and where there was clear lines between the enemy, the other, and the comrades in solidarity. What you saw in a certain moment was how the line separating enemy and ally dissolved, and how the HIV activists reached into corporations, reached into the government, and created some of these very personal relationships and personal alliances with people in power. So there's a lot of good cultural products out there. There's the film The Parasite, which came out a few years ago, which characterizes this impoverished couple, this family that's living in the family of a rich, wealthy elite. I think there's all kinds of really good histories of figuration related to the parasite. There's a book by Michel Serres called The Parasite that is a really playful and poetic and philosophical exploration of the tensions between the guests and the hosts. And it's a difficult book to teach. I've taught it before. Students often try to puzzle through these very contradictory and playful ways of using language. But if you think about the parasite as a biological or social freeloader, and the landlord could be a parasite, a billionaire could be a parasite. You can think about the new, in this era of uncertainty, the new regimes of excess and parasitic exploitation of certain kinds of capitalists. I think this is where the figure of the virus can be really interesting to think with and beyond the parasite. So in conventional textbooks about virology or biology, the parasite is the virus is the irredeemable other, the parasite that should be eliminated. But the new virology is about accepting the fact of coexistence, accepting even if these agents are interrupting stable processes, getting into organisms, sometimes causing a cough, sometimes causing a disease. It's about how to not eliminate these categories of being from the world, but imagine living in contingent relationships in happiness and harmony. So this isn't a talk saying you shouldn't get vaccinated, you shouldn't, take the therapies that are available to you for the cold or flu, that you shouldn't mask. Like all of these biomedical knowledges are, in my mind, essential for living with viruses in happy, healthy, harmonious ways. But maybe one of the things you can take away is how to get past the anxiety and uncertainty about viral infections. And maybe it's a way of rethinking a disposition to the world in this broader time of anxiety where we have warfare, where we have new regimes of economic exploitation. So if we can rethink our relationships with viruses, we can maybe rethink our social relations, transforming those, again, antagonistic relations in social and political worlds into harmonious ones, not necessarily ones where power relationships disappear. I don't think the corporation or the nation state or any kinds of hegemonic political entities are going away anytime soon. But maybe how do we become better parasites ourselves, working within these institutions, working within these structural forms to transform them from within?
Yeah, just want to say something here. I think it's interesting to hear that when we talk about peace and people are mentioning warfare and ceasefire, especially at this particular moment when we have conflict on the Thai-Cambodian borders. I think one way of thinking about peace is to distinguish between negative peace and positive peace, which is among peace study scholars, has been a very, very important concept to work around this confrontational way of looking at peace is the state without war, or the being able to keep to be separate without any interaction with the antagonistic relationship. So I just want to mention that. But my question to you is that when we listen to this biological world of diverse being, and it seems that virus itself is probably, I want to just ask if the category of virus itself is still useful, because we are having all kind of biological being like virus, macrophage, or even some kind of protein like prion, which is not necessarily living, but it can still be pathogenic and also reproduce itself, and up to all kind of other life form, which may be in between these categories. So my question is that to what extent the category of virus itself is still useful. This also relates to my next questions about viewing human being as a biological assemblage of all kind of being like microbiome, and now you have this virome thing. Is this view of looking at human bodies as consisting of all kind of biological being, is it still a very much anthropocentric view?
So I loved those three nested comments and questions, and maybe to think about the first. Certain kinds of peace are hegemonic. They're enforcing exploitative relationships. There's a really good book by Bruno Latour. I think it came out around 2002, called War of the Worlds, right after September 11th. Latour was insisting sometimes it's better to be at war and recognize the very difficult political and diplomatic work that needs to be done rather than pretend that you live in this state of peace. So I think, at a moment when U.S. hegemony is coming unraveled, we can see all the violences embedded in earlier regimes of imperialism and exploitative global relations of capital. Maybe on an abstract level, you could think about those kinds of persistent pieces that might not be a good piece in relation to the stable state of a human organism in the face of, in some ways, the COVID-19 pandemic forced us to rethink modernity, forced us to rethink, to some extent, the kinds of exploitative social and labor relations that were sustaining society, but also led to some soul searching about the food system, global systems of travel, things that were taken for granted that were naturalized. Let's see, thinking back to the other, so quickly remind me the other two. There was three really interesting things. So peace. Yeah, okay, so viruses in many ways, you're right, is a total junk category. So it's not a taxonomic category like primate. You can't trace a single point of origin for viruses. You have RNA viruses, you have DNA viruses. They're totally unrelated. So, and now remember the last part of the question. So is there a risk of anthropocentrism in this? And I say, yes, absolutely. So I did a PhD defense last week in Copenhagen, where the candidate was very interested in the holobiont, this other concept. So the holobiont means the whole organism. So that's if you're thinking about a human or a plant or another organism, there's kind of the so-called human cells or the so-called plant cells, but there's all the microbes that exist within and around. So one of the questions I posed to the candidate is, can you think about the human as a holobiont, but also as the bacterium, like a singular bacterium with viral infections as a holobiont, and the viruses as kind of a transient set of connections amongst holobionts. So there definitely is a risk of naturalizing the human when we're thinking about all these relations and to be fair, we're mostly anthropologists and sociologists in this room, so to a certain degree anthropocentrism is justified, at least in this room. But if we try to rethink the human from the bacterial viral standpoint, how might we rethink our set of interests, our set of concerns, not just from keeping the stability of healthy peace of the human body, but a multi-species dynamic assemblage where symbiosis in the gut is contingent on these constant exchanges amongst not organisms but holobionts. I think maybe that's the way to get beyond the latent anthropocentrism that might result in some pathologies in human health. So if we're relentlessly thinking about the human as a border, a bounded object with boundaries that need to be policed, if we're using antibiotic logics rather than probiotic logics, I think maybe that conceptual move that you're pointing towards of getting past thinking the human organism is the whole organism, but thinking the human in relation to other holobionts, I think that's maybe where we should go.
Thank you. I think we have only, it's 10:45. We are on time. So maybe just one last question and we can continue throughout the break then. Yeah.
Thank you, Eben. I feel like an adult meeting a rock stars. I'm so excited. So my name is Piyatev. I'm SCS anthropologist. So my question is, we know from your presentation about virus, the agency of virus in multi-species studies. Then how about the agency of technology because when we, in this photo, we can see that we with our own human eyes we cannot see, we cannot know the existence of the microbial species, but we use the tools or technologies to know, to get in touch with them. Then how about the influence or how technology entangle with this assemblage?
Yeah, I think that's a really good and important question. So, for starters, I think it's appreciating the limits of technology at any particular historical moment. So, if we think back to 25 years ago, when virologists thought that they knew the world, they were relying on electron microscopes, which produced those fuzzy black and white pictures. They were relying on a very crude assay called the viral plaque assay. You basically have a cell culture in a petri dish. You apply a virus, you see how many cells die. That gives you sort of an image of the presence of a virulent virus. So that was the earlier technological assemblage that in the 70s, 80s, 90s produced this sense of like, oh, we know all the viruses. We appreciate the world of viruses, and we more or less have it figured out. Bruno Latour and Isabel Stingers describe facts as factishes, fetish objects that obscure the unknown. So you think that you have, at any particular historical moment, technology and technoscience gives you this stable world. We have the sense that, there is a known universe of facts, but when you push at one of those facts and suddenly it breaks, or suddenly a new technological apparatus comes along, like bioinformatics, that genomics and genetics, that gave us a new way of apprehending viruses, you really appreciate how what we might call the technological arts of noticing are really foundational to the world. So if we think of the modern world as this contingent assemblage of technology, of factishes, and we're starting to see a whole, not only mode of knowing, so technology enables us to know certain things, but it's also, that's epistemology, but it's also, modernity is an ontological project. And the technology does things. So we could think ontology and epistemology together as structuring the known world. Right now, and this is speaking to the theme of the conference, that onto-epistemology, sustaining the modern world, sustaining this amazing building, the food we're going to eat today, the electricity, we can appreciate how fragile it is at this moment. We've got climate change, we've got biodiversity crises, we've got the specter of new pandemic diseases. So there's certain things that our onto-epistemology knows well and does well, but beyond the purview of our current technoscientific apparatuses, there's certain things that are escaping. There's certain things that we know less well. And we might be able to appreciate how these structures of knowledge and structures of being and doing, the infrastructures are starting to crack and fail and erode. But I don't think we're quite there yet to think through what comes next. And I think maybe that's what the rest of the presentations here today are going to do in very situated terms. What kinds of cultural knowledges, what kinds of political and economic assemblages, what kinds of multi-species and ecological assemblages must be built and reconfigured now to address the failings of this very fragile modern onto-epistemology.
So I think the debate is getting hotter and hotter and more and more engaging. But actually, we are a little bit behind the time, and maybe we could just continue, as I said, throughout the break. So thank you so much, Eben Kirksey, for very thought-provoking